All right, everyone. We're going to have a talk here. It's going to be, we're going to talk about vaccines and immune disorders. But the first half, we're just going to hit the vaccine side of it at this point, and we're really going to talk about it in comparison to the cancer vaccines that we just finished our talk on. Now, so that's really kind of be the first half. The second half of the lecture will go into the immune disorders. Okay, so they're really, when we talk about vaccinations, immunizations, there are really two types of immunizations, and that's you can have active or passive immunizations. Now, passive, we won't spend a lot of time talking about it, but the basic idea of passive immunizations is that you're just administering antibodies, so purified antibodies, not necessarily from the same organisms. They could be human or horse or depending upon the treatment. But they are short-term. They provide kind of an immune boost, but they don't create memory cells. Active immunizations are creating long-term responses, and they're really this is what we're kind of most familiar with, whether we're getting diphtheria, smallpox, those types of things. Those types of vaccinations create memory B cells, and as I mentioned with the cancer vaccines, what we're going to be talking about with these types of vaccinations are development of the B cell response. Cancer vaccines are working on T cell responses. So these are B cells, and cancer cells are T cells. So keep that in mind. Now, what you're seeing to create these active immune responses, there are really different ways you can do it. You can have natural infections. For example, if you were a kid and one of your friends got chickenpox, I remember my mom sent me down to my friend's house, and I had to hang out and play until I got the chickenpox. There are the more traditional ones are taking the pathogenic organisms, viruses, and you attenuate them, weaken them, or inactivate them. Or you can take pieces of them, and we'll talk about some of these strategies later on, that will create immune response without exposing the people to the actual infectious agent, even though in this case they're inactivated or dead. Passive treatments, as I mentioned, short term. So here is a list, and it gives you a flavor of the types of things that you can get for polyclonal antibodies to cytomegalovirus. You can get horse antibodies, human or horse antibodies, for rabies. For snake bites, there's horse antivenom. Tetanus, again, you can get horse antitoxins. It reinforces the idea that I've already mentioned. Again, these are short-term responses. These are treating people with antibodies such that they then get short-term immune response defenses against these particular things. A lot of these are given to people when you're traveling to countries that have a lot of disease, if it's hepatitis A or B or things like that, where you might be exposed to things that you're not commonly exposed to, and so you don't have a robust immune system for them. So you'll hear things like getting gamma globulin shots or things like that to help kind of boost up your immune system and protect you against the types of exposures to pathogens that you might see in those countries that you're not normally seeing in America. Now, again, short term. So now, what about the long term? So these are probably more what you're familiar with, whether it's polio, measles, mumps. You're getting things like diphtheria. I think you can get flu viruses, flu things. These are yearly, and even though we said they're long term, it's not that your immune response is short term. You can get it's the flu virus, as we've talked about, can change so rapidly that you need a new vaccination every year. So these are the types of things. These are looking at mostly with kids, what happens when your immune system is developing. How do we actually help protect them? Now, what are the different types of vaccines that are used for these treatments? So here, and I mentioned this earlier, this is from that previous chart where you can look at attenuated, inactivated, or killed. So these are using the entire pathogenic organism, whether it's a bacteria or a virus, that is inactivated, killed, or alive. Attenuated means it's weakened. So attenuated means, yes, you are getting the actual infectious agent, but it's weakened to a point where your immune system will be able to mount a response and prevent any infections by the actual virus. Now, the benefits of these things, as you notice here, if we look at it, the closer you get to the actual infectious agent, the better your immune response, the better the memory cells, the development of memory cells are. So you can see, so you can see that those are the advantages. Now, the problem is in terms of there are issues in terms of refrigeration or storing these, and so that can be particularly something when you're transporting the actual vaccinations, especially to countries that don't have really well-developed medical systems, or that the closer it is to the actual live virus, there can be mutations and it actually can create a virulent form. So the farther you get away from the live or actual pathogen, the safer it is in terms of its ability to actually cause an infection. Now, so that's what you see as we go down this chart. There are different ways. What we're doing is making vaccinations that are farther away from the actual pathogen. And so in some of these toxoid subunits, conjugates, DNA, recombinant vectors, some of these are not yet available. But the idea is you're going to take, and we're going to go into looking at these types of things, you're going to take antigens from the particular infectious agent and then develop a vaccination that will expose you to that antigen, never exposed to the actual pathogenic agent, and so they become safer. They're harder to develop, however, and so there are some pros and cons to these things. Okay, so let's talk a little bit about these particular subunit vaccines. So toxoid and subunit vaccines are only using pieces of the pathogen, and so what you're doing is trying to create a vaccination with these small parts, something that you feel confident or know that is an antigenic part of the pathogen, that your immune response will create an effective defense. Will create an effective defense against that particular pathogen. So let's look at bacterial toxoid viruses are actually used with a bacterial toxin. So in many cases, bacteria like pertussis, that's the bacterial toxin that actually is so damaging. Diphtheria, as well, that you can actually conjugate or inactivate the bacterial toxin with formaldehyde, so it's kind of like an attenuated virus or heat inactivated vaccination. But in that case, it's just the bacterial toxin. You're not getting the actual organism. Subunit vaccines, you're picking and choosing different parts of the proteins. For example, you can take the capsule or membrane polysaccharides, glycoproteins that are on the coat proteins of a virus. And subunit vaccines, actually one thing we can talk about a little bit more, but there are actually genetically modified plants right now that they're genetically modified to produce proteins from the pathogen. A classic example is there's a banana vaccine out there in which part of a hepatitis virus, I think hepatitis C, is incorporated into the banana genome, and so the idea is that it's expressing part of this in the flesh of the banana, and so every time a person eats or is exposed to it, you're actually getting a small bit of vaccination of that antigen, and that will help produce and kind of a an amount of memory B cells, which will kind of produce your response and protect you from the actual pathogen. So here's an example of some of these conjugate vaccines. So conjugate or kind of subunit vaccines, in this case, they're taking the polysaccharide subunit. So all they've taken is, this is the polysaccharide, so the polysaccharide that's on the cell membrane. So this is our pathogen here. And if you take the coat protein, these are good targets for these are good targets for creating these kind of subunit vaccines, because these coat proteins are obviously some of the things that are going to be most prominently displayed to your immune system. So creating vaccines with these viruses is a good bet, so to speak. In this particular case, you can produce IgMs and memory cells. So it's not the best. You don't get a full immune response, but it's a much safer that you're not being exposed to a particular pathogen. Now, one thing that's kind of an interesting idea is that if you conjugate, so if you covalently conjugate a toxoid, so remember that is the formaldehyde inhibited bacterial toxin. This can actually stimulate thymus dependent pathways, and now you're getting memory B cells. So really, kind of the idea here is, remember these IgMs. This is the thymus independent pathway. So thymus, if you remember our T cell maturation lectures, when thymus independent, you have these B cells that don't go without T cells, without the help of T helper cells. You don't produce memory cells. You don't go through class switching, and so based on that, with these polysaccharide coatings, you get kind of a limited immune response. By kicking in this toxoid, it creates an antigen that will now stimulate thymus dependent. You use T helper cells. Those T helper cells are going to produce the cytokines that will stimulate plasma cells differentiating different classes. You get all the memory cells, and so you get a stronger vaccine. So it's kind of a trick to get things into this thymus dependent pathway. Multivalent vaccines are another type of, way to increase your immune response, and so in this case, the problem is with these. How do you, if you want to kind of create safer vaccines, right? So in that particular case, you're getting away from, okay, let's not have vaccines that use the whole virus, particularly if that virus is a very damaging, very dangerous virus, and that in particular cases that you can't, if you heat and activate it, it damages it so greatly that you don't get a immune response that's useful or relevant to the particular live virus. So in this particular case, you're trying to find recombinant proteins, pieces of, let's say, a virus, something like that, that you feel will elicit immune response from your immune system to that particular viral pathogen. So in this particular case, what they've done is create this mechanism to conjugate antibodies to a solid matrix. And you pick some epitopes. So these are your recombinant proteins here that you think are going to be good epitopes to stimulate immune response. And so the ISCOM is a mechanism. It's kind of you create a little membrane-bound complex, these kind of almost kind of fake vesicles. And these vesicles will will fuse to cell membranes. It's kind of like the transfections, kind of like the liposomes that I think you're using in cell culture. That this is a way you can deliver kind of this protein target to the cell membrane. So then once these guys get into, sorry, I'm kind of jumping ahead here. So once these guys get in, you fuse the cell is, these peptides are integrated or go into the cytosol. They're ground up by the proteasome here with that LMP 7 and 9 subunit. Those guys will create peptides that are expressed on the ER in MHC class 1 receptors. So now this becomes an interesting way to get viral peptides because those are the things you're looking for, or maybe even cancer peptides that are expressed in MHC class 1 receptors and then develop an immune response to that particular antigen because it will be recognized as a foreign antigen. The solid matrix, so this is another way of getting in and presenting peptides so on antigen presenting cells. So the FC receptors will facilitate phagocytosis. I kind of jumped between those two. But you have kind of two different ideas, two different ways of delivering recombinant peptides. One, hooking them up to antibodies. The FC receptors on macrophages will induce phagocytosis, present these things on antigen presenting cells, present these peptides, things that you think will induce an immune response. Another way, through these kind of vesicles, liposome type transport vesicles, to get peptides that are presenting on MHC class 1 receptors. So those are kind of two separate techniques for getting antigens or epitopes presented on cells. Another interesting way to do this is to create DNA vaccines. And this is a very intriguing idea, meaning instead of figuring out, as we did on that last one, kind of difficult way, or not difficult, kind of tricky ways to get peptides into the immune system, which can be challenging, especially if you're trying to create a vaccine that can reach huge numbers of people, especially if they're in third world countries. You know, if you're like the Gates Foundation that's trying to create malaria vaccines that will go into African and very poor countries that don't have established medical distribution, kind of the framework to do that. Well, you know, it can be challenging to distribute those things. So one way around it, or one potential way around it, are these DNA vaccines. And DNA vaccines are again doing the same idea as what we talked about. You're trying to get epitopes. You are cloning out antigenic proteins from a particular pathogen, and you really target, you know, you'll target sections of it that are sections of this pathogenic organisms that you feel will be highly antigenic. So when you clone those out, you can put them into a mammalian vector. And what you're trying to do is just get that DNA. So all you have to do is get that DNA you're trying to integrate that DNA into the genome of cells, muscle cells and dendritic cells in an area of injection. Okay? So here's our particular plasmid vaccine. And so in this case, you're trying to prime the cellular response, meaning you put that into antigen presenting cells. You can get, or into normal muscle cells, they can present this kind of this antigen on MHC class 1 receptors, and you'll make cytotoxic T cells that will then create a T cell response and you know, an immunity to that particular vaccine. If you get it, if you get them into antigen presenting cells, you get this vaccine. APCs will present antigens, and then you can create the other side. You can get B cell responses as well. So in this case, if this, what it's showing here, you can present, you have your antigen integrated into the host genome here. It creates antigenic proteins presented on your MHC class I receptor. These peptides, you can get essentially both sides of the immune response infecting, especially if you infect antigen-presenting cells as well. So you can get MHC class I and II cells working, and you get essentially both sides of your immune response working. It works well in theory. There aren't a lot of examples of these working in practice yet, but the big advantage of this is you can create vaccines that are very cheap. It's just cloning. Once you have the plasmid, you just have to grow up a bunch of the plasmid, do a bunch of DNA purifications, and then just take it to wherever you want and do the transfections, inject it into muscle areas, and you'll hit both B and T cells, or both sides of response, because you're not directly targeting, you're just essentially getting random integration into a bunch of different cell types that you're going to activate B and T cells in this response. That's in theory. Now, one challenge is, you're only using one type of antigen. So if your particular pathogen mutates or changes, you won't have immunity to it if that particular antigen changes. Again, you're not going to get the full polyclonal response that a particular pathogen will have. That's what makes your immune system really robust, is when you get a polyclonal response to the dozens and dozens or hundreds or thousands of different antigens that are on a particular virus or bacteria. In this case, you're choosing one, and you're kind of hoping that one is going to be the best one. Now let's look a little bit in detail of kind of how you can do recombinant viral vectors. Now, in the previous one, if we looked at DNA vectors are very general, and the idea is you're injecting them into tissue, you get random integrations, you're hoping to hit muscle cells, you're hoping to hit antigen-presenting cells, you stimulate B and T cell responses. Recombinant viral vectors are different because they would be targeted, meaning they would target tissues that you want this antigen to be, expressed and presenting. There's not the randomness that a DNA vaccine would have. So in this particular case, you're going to do the same thing, though. You're going to clone a particular gene for an antigen, and you get it into a plasmid. In this particular case, we're going to use the vaccinia virus as our host. Now, the vaccinia virus is not something, it is pathogenic, so you don't want to give somebody a live vaccinia virus. So you have to kind of come up with a technique to create a recombinant vaccinia virus. In this particular case, it's kind of an interesting idea, which is you take that gene and put it into a plasmid, and you insert it into this TK gene. And TK gene is a gene that's used a lot in mammalian cell culture. It's thymidine kinase, and it's done that because you can have a suicide selection for that gene. So cells that express thymidine kinase will die when you add BUDR, and that's the BUDR selection. So what you're doing is you're looking for kind of co-transfection. You take tissue culture cells and infect them with the virus. You infect them with transfect them with your plasmid. You're going to get recombination between the virus and the plasmid at the thymidine kinase gene. So the vaccinia virus has that. So when you get a recombinant virus, which is what you see here, the homologous recombination between the thymidine kinase gene here and the one here will essentially put this gene from the pathogen, and that inactivates the thymidine kinase because you have another gene right in the middle of it. So the thymidine kinase gene in the virus doesn't work anymore. So any intact virus dies here. So when you do this, the BRDU selection kills intact vaccinia viruses. What you're left with is a recombinant virus that is going to express your gene of interest. And so now when you infect a patient with that recombinant virus, you get very efficient transformations, meaning these viruses are meant to infect cells and integrate that DNA. It's not the random nature of the DNA vaccines where you just inject and hope for expression. Not all cell types will get hit. Maybe it won't work as well as you want. Here, you can be confident this vaccine is going to infect cells and deliver your DNA, and its job is to recombine into the host genome. That doesn't happen necessarily with your transfection with the DNA virus. So the advantage again is you're going to get higher transfections, you're going to get better expression. Disadvantage again, it's expensive. It's a lot more work. And then you are again betting on a single antigen, just as we were with the DNA virus. You're betting on a single antigen. If your virus or your pathogen changes, mutates, you can lose some of your immune defense for that particular pathogen. So it's expensive, it takes a lot of work. If your pathogen mutates, changes, then there's a big problem. Then you have to start over again. Now what we'll do in class is kind of go through this, and we'll talk about different ways if we wanted to create a vaccine for swine flu. We'll go through and I'll assign each group one of these different ways, and we'll talk in class about how you would make or what are the advantages and disadvantages, and how you would make of attenuated virus subunit vaccines and DNA vaccines, recombinant vaccines. We'll talk about that in class next week.